Hello students, today I will introduce the general aspect of mutagen. I will talk about the general aspect of mutagen, physical mutagen, biological mutagen as well as chemical mutagen. I will talk about their application, their resistance, protection as well as effect over the environment and general effect. As introduction we can say mutagen directly affect the DNA, directly damage them off. They can change the insertion or the replication forms, okay, and they can cause the replicational error, chromosomal separation. Some mutagen are pro-mutagen which by metabolism can be converted to the mutagen. So, mutagen is the agent by which the mutation can cause. When we are talking about the background, the spontaneous mutagenesis happens by replicational error and spontaneous hydrolysis, but induced mutagenesis happen by the exposure to the mutagen. Next, the historical background. In 1927, by Muller, the effect of X-ray was found over the fruit flies Drosophila. Then in 1928, Altenberg found the effect of UV radiation over the fruit fly. In 1928, Stadler found the effect of X-ray in barley, and in 1936 he found the effect of UV radiation over the maize, and in 1940 Charlotte Auerbach found the effect of mustard gas on fruit flies. Characteristics. So, it can introduce the frequency of mutation, okay, and it actually imparts some change in the coding system. It can change the base pair, it can induce the mutation, and ultimately the observable phenotypic change can be monitored. Next, classification and types. There are so many classification of mutagen. Number 1 physical, number 2 chemical, and number 3 is biological. Within physical mutagen, we can find heat, microwave, radiation. Within radiation, particulate and electromagnetic radiation is found. Electromagnetic radiation may be ionizing as X-ray gamma ray or may be non-ionizing as UV ray. Chemical mutagen may be the base analog, DNA interacting agent as well as chemical modifying agent. Biological mutagen may be transposon, virus or bacteria. Mode of action. So, it can change the chromosomal abnormalities by inversion, deletion, etc. and it can change the base modification by insertion, deletion as well as substitution. Ultimately, it can change the structure of the protein, but sometimes it is not changed by the codon redundancy, but it is actually changed some sort of chromosomal number as well as aneuploidy. Next, the strategies. Strategies may be general or main. As general strategies, we can say it can cause lethality, it can cause death, it can cause cancer, loss of gene function. General biological effect. It includes that by X-ray and any other ray, the ionization or free radicals are produced. It can damage lipid, DNA as well as other macromolecules. And we can find it can lead to the cell death, the impairment of the cell division, arrest cell cycle as well as it can cause damage to the bone marrow cell and gastrointestinal lining cell. As general aspect, we can say it can produce the ionization which is responsible for radiation mutagenesis and radiation genetics. Radiation mutagenesis. It includes some phenomena, factors affecting the radiation efficiency, then effect in different system and some dosage kinetics including dosage mutation relationship. First one, phenomena. I will talk about the photoelectric effect, Compton scattering and pair production. So, photoelectric effect is that sometimes the low energy photon can help to eliminate some high energy radiation. So, these secondary electrons are kicked off from the orbit having higher damaging effect to the biological molecule. Compton scattering, sometimes high energy photon can help to radiate some other wavelength of radiation having the low energy and wavelength is much more higher. Pair production. It was found that when radioactive nuclei absorb some radiation, it gives rise to some one electron and one positron. So, the two ions are getting produced. The factors affecting the efficiency of radiation including temperature, oxygen concentration, cellular factor, aging, mechanical stress and chemical component. First one, oxygen. Now, oxygen tension and this sort of concentration was found in case of different systems as well as involving mechanism. Now, in different system means we can find that in Visia faba, the low oxygen concentration give rise to low mutation rate in recessive lethal. We can find in Drosophila 4000 Rontgen unit increase 14 to 18% mutation rate with the presence of oxygen. It was found that in spermatid, the higher oxygen concentration than spermatozoa, showing much more chromosomal damage. Next one, mechanism of oxygen. As we know that the H2O is broken to the free radicals along with oxygen is giving rise to hydroperoxyl radical as well as hydrogen peroxide. This hydroperoxyl radical and hydrogen peroxide is much more devastating towards the biological material. Next one, the effect of temperature. Now, very high and very low temperature shock can increase the irradiation related mutation, and it is actually influenced by the position effect. Next one, aging effect. We can find in case of snapdragon the increase in 14% of mutation rate in 5 to 10 years old seeds, as we can find in Drosophila the 2 to 3 weeks aged sperm show higher mutation frequency towards the radiation. Next, we can also find the sex-linked lethals are increased in Drosophila, and this aging related damage impaired if in mammal system causes cocaine syndrome. Next one, cellular factor. Mutated gene increases the mutation frequency as well as position effects can influence the positioning of the gene as well as effect towards the irradiation. Because near the centromere, much more chromosomal breakage happens. Chromosomal breakage cannot be reverted, and in case of mice, the 7 loci are found which actually act differentially to the same irradiation. That is called mice 7 locus test. Next one, the mechanical stress, sonic vibration and centrifugal force can give rise to more chromosomal break. Next one, chemical component. The presence of the colchicine, which actually impairs spindle fiber formation, which also impair the chromosome break, that is the reduction of the mutation frequency. And cyanide actually inhibit the cellular respiration, so more oxygen is available for non-metabolic purpose to give rise to reactive oxygen species for radiation breakage towards the DNA. The effect of radiation in different systems. In Trillium species, sparrow found the chromosome breakage is 60 times in metaphase than that of the interphase, and non-dividing cell do not respond much more than that of the dividing cell. Target theory, consequence as well as mathematical expression. Target theory. It states that the gene is the target and the radiation is the hit, and by single hit point mutation can be caused, which is showing linear relationship with survival fraction and dose, and we can find the exponential relation with that of gene mutation. But in case of chromosome break, for 2 hits, the square of the dose is needed and the kinetic is geometrical. For multiple hits also, the geometrical kinetics is applicable and much more hits are needed. In case of Drosophila, it was found that the chromosome break occurs before DNA synthesis and chromatid break occurs after DNA synthesis, and it was found that the sister chromosome repair system is actually taking place within the course of the irradiation by healing system, and it was found that the quality of the radiation is dependent on the density of the ions as well as rate of the mutation. As consequence, we can also say there are 15 to 20 hits are needed for the chromosome break, but 1 hit is needed for the sex-linked recessive lethal, and the mutation frequency is independent to the radiation intensity, and it depends upon the maturation state of the cell of Drosophila, as well as it depends upon the sensitivity of the radiation to the cell as well as the tolerance to the radiation. If dn number of the cells are getting damaged with dd dose, which is proportional with the n number of cells which are not yet exposed. So, minus dn by dd is equal to Kn, and survival fraction S is equal to n by n0 is equal to e to the power minus Kd. The observation curve. It was found that if n numbers of sites are there in an organism of a population, then the n hits are needed. So that is called n hit kinetics, and ln S is equal to ln n minus Kd. If the target volume is V, then S is equal to e to the power minus CVD. It was found that in case of organism, if nucleotide content is double than that of the other organism, so that initial organism is the double time sensitive towards the irradiation and it needs half dose to be affected same. Next, in different system we can find the application of this target theory. In case of bacteriophage, we can find this sort of n-hit curves give different types according to the nucleotide content of different phage. In case of yeast, it was found that haploid diploid condition. Haploid condition is affected by single hit and will be impaired to form colony. But in case of bacteria, as during replication, the chromosomes are separated. So by single hit, the colony formation impairment cannot be found. It includes the breaks of the DNA, cross-linking as well as base damage or loss. Next one, biological indication. Bacteria will be unable to form colony by irradiation, phages will be unable to form plaques, and single-stranded breaks are found in case of microorganism as well as double-stranded break. Single-stranded break can be ligated by the ligase, but double-stranded breaks are lethal, they cannot be ligated by the ligase, and it was found several other base damages also. Next one, survival curve. So survival curve is actually found in case of UV radiation. The dose dependent curve with respect to the radiation is called survival curve, where we can find in case of bacteria the first portion is not affected, that is linear. That means that is not yet affected by the radiation, then it is getting declined, and it is actually affected by the environmental factor and the repair mechanism of the internal cellular processes. Next one, dose of the radiation. It actually includes the unit of radiation as well as the measurement process and effects in different systems. Next, unit of radiation. The first one, Rontgen unit. Rontgen unit is the dose of X-ray or gamma ray which is able to produce the ion pairs having one ESU charge for each ions, and that is happen within the 1 cc of the air. Next one is RAD, relative effectiveness, that is radiation absorbed dose, and it was found that this radiation absorbed dose is differentiated, and it is the intensity of the irradiation that is found carrying the charge, that is 100 args per gram. RBE, relative biological effectiveness, it was found that the dose of the 200 kilo electron volt X-ray to show specific effect divided by dose of other radiation to show such specific effect. REM, Rontgen equivalent mean 1 rem of all the radiation can create the same effect. Now the correlation, that is the RAD into RBE is equal to REM. As general aspect, I will talk about the different types, the mode of action as well as the test to assess the mode of action. Number 1, base modification. So it is done by the base analogues. Number 2, the DNA interacting agent as DNA intercalating agent like proflavin, cross-linker, and the chemical modifier as deaminating agent, alkylating agent, etc. So as mode of action, it can show frame shift, bulky adduct formation, mismatches, insertion, deletion, and the halt of replication and transcription. To test the mechanism, the bacteriophage T4R2 locus is taken to show the forward as well as reversion to produce the normal R+. By 5-bromouracil base analog and alkylating agent which can cause some sort of transition, it actually can be reverted easily, transition or by transversion. But by proflavin as intercalating agent which actually causes insertion or deletion cannot actually revert this towards the backward mutation to normal R plus loci. So, as general aspect, we can say papilloma virus can cause cancer. The stomach cancer is caused by bacteria, that is Helicobacter pylori, and transposon causes insertional mutagenesis. As application, we will talk about in case of plant, that is mutation breeding, and in case of animal, that is cancer therapy. The mutagens can be taken for the cancer therapy. Daunorubicin, cisplatin are the chemical mutagen can be acted as the cancer therapy, and the radiation are taken for the radiation therapy. Next one, in plant system, Z-maize and Arabidopsis thaliana are taken to assess the mutagen. Animal system, number 1, cell culture. Chinese hamster ovarian cells are taken to assess the bacterial mutagenicity and for other purposes. Number 2, the chromosome check system, because the sister chromosome are interchanged during the course of the application of mutagen, so they can be tested for the mutagen assay. And the third one is animal test system. Actually, the rodents are taken for this purpose, and the foods and water are actually supplied with the mutagen. Sometimes it can be applied over the skins also. And in some cases, the transgenic mice are taken for vectorial system to assay the mutagen system. So, protection can be done by several antioxidants, by vitamin C, vitamin A, tocopherol, beta-carotene, etc. And selenoprotein and glutathione peroxidase are also accumulated here to show the effect. And the induction of cytochrome C450 is also give the protection against mutagen. Next one, mutagen and environment. It was found by some researchers that 3 genera, 27 species and 13 subspecies of the fishes were abolished due to the chemical pollutions. And the PCB causes mutation in the mini satellite genes. And we know that the UVB rays are coming via the ozone holes, which can create the genetic variability to sustain such sort of mutational environmental damage. The resistance against 8-azaguanine was found in Salmonella typhimurium. The resistance against 5-methyltryptophan was found in E. coli, and the resistance against mammalian system was found against obein, and it was tested by OUA assay. And the mutation or resistance against 8-azaguanine or 6-thioguanine was found by HPRT assay system, that is hypoxanthine phosphoribosyl transferase system assay. Then the repair of the DNA damage causing by the mutagen. So, there are some of the repair mechanism within the cell, some of the enzymes by which the damage caused by the mutagen can be repaired. So, viewers, I have talked about different types of mutagen, their mode of action, their effect, as well as the impact over the environment on several aspects. The environment impact is negative, so thus mutagen can cause as carcinogen as cancer, so they can be avoided. Thank you.